adjust the mic so that you hear me well in the front and also in the back seat. Do you hear me well in the back seat? Good. All right, ladies and gentlemen, greetings and welcome to another chapter of human physiology, this time on reproductive and sexual function. Finally, this is one of the most intriguing parts in the course of the year. This lecture series comprises 4 consecutive lectures and will span around 2 weeks, given that we will have the spring break in between. So I must say, first of all, I'm really delighted to see that you attended in vast numbers despite the late hour and despite of the approaching spring break. So the first 2 lectures will be delivered by me. My name is Semmel. I work also at the Department of Physiology. And I will talk you through the male sexual reproductive functions, and also we will touch on the sexual differentiation in humans. And then Professor Fitt will come to stage and will talk about female reproductive functions with special emphasis also on contraception, and then delivery, childbearing, lactation, and everything that has to do with those functions. So, in the first 2 lectures, you'll be having me, that's that. And there will be a little twist as compared to the original schedule or to the official schedule that you might have seen already at the website, because we will start with the physiology of male reproductive functions, and only that will we discuss differentiation, all right? So there is a swap between the first and the second lecture. All right, shall we begin? Thank you, Shab. I think we should. I know that by now you're probably fed up with the fact that every lecture series starts with a statement that the topic you're about to hear is really the most important. And then just really give you numbers and statistics and everything that's supposed to be interesting, yet it's just boring. Now let me please be just really true to that tradition and give you some numbers. Here they come. First of all, this is the predicted population of the Earth, and we are right here, around 7 billion. And there are quite a few estimates as to the growth of the population in the next decades. Most experts say that you can probably neglect the minimalistic predictions, and the population will grow according to the yellow, or for all we know, it could also grow exponentially according to the red line. It would be probably just very easy and simplistic to conclude right here that, oh my God, we're going to overpopulate the planet, we're all going to starve to death. Having said that, this may still happen. However, let's look at these numbers with a bit more detail and putting them for all into the geographical context. What you see here is the distribution of population growth between different countries and continents in the world. And what may be very apparent that there's a certain dichotomy in the numbers. Some countries, mostly in the northern hemisphere, do not display any significant growth in their population between these dates, and there are other countries, mainly in the tropical regions of the Earth, where the population grows rather quickly. That's really not a surprise, but if you take this data and merge it, or rather superimpose the data of expected lifespan, then you or we may see something more disturbing, maybe I should say, but I wouldn't go as far as to say that. So if you superimpose the 2 data, what you see here is where the population growth is the highest. There we have the lowest life expectancy. So So from the medical point of view, it's not necessarily, the problem is not necessarily the growing population. Actually having more fellow humans should be a happy thing, shouldn't it? The problem is that there are quite a few millions to come, and what we cannot do is providing them a proper life quality and expectancy. And that's now a more serious issue, isn't it? So, but there are two sides to this actually. So where some parts of the world some families may have more children than they originally intended to, the neighbor, the couple in the neighborhood may face problem conceiving one child in the first place. And this is the other side, the other aspect of the human fertility, or rather I should say infertility. Infertility is such a hot topic nowadays that the World Health Organization predicts infertility to become the third most serious medical issue in this century. Third one, at least in developed countries, they predict that 1 in 6 couples will face fertility issues at some point during their relationship. That is an incredibly high rate of predicted infertility. As a matter of fact, it is directly comparable to the frequency of stroke or prostate cancer. Now, we all know what kind of burden these two diseases mean. Then we can have an idea as to what burden fertility will put on society. There's another aspect, yet another aspect, and that's the aspect of infectious diseases. Human sexual intercourse is a channel through which pathogens can infect yet or currently healthy individuals. There are around 500 million new STDs, curable new STDs every year. These are the documented cases. 500 million, that's also a mind-boggling number. But the question is, do these numbers in the 21st century, where the awareness of STDs is actually rather high, do these numbers make us more precautious or less precautious, or we just don't get second hoot about these? I won't tell you which country these numbers originate from, but these are youngsters, adolescents, in their during their what's that called, high school period, and the entire column shows the percentage of people already having more or less active sexual life, right? And the blue one is the part of this population which actually is willing to use condoms. And as you can see, the older they get, obviously the more people engage in sexual activity. The problem is the population, the purple one, who is willing to use condoms, is not growing. So the older we get, the more confident we are that that will not happen to us. And actually these numbers, the willing to use condoms is even worse between the age of 20 and 40. And also, and this will really underline the importance of education and precaution and medication as well, that nowadays it is estimated that around 1/3 of new HIV infections happen during childbearing or breastfeeding. And the problem with that is that these are cases which could have been avoided with proper awareness, medication, and education of the population. And you, as future healthcare professionals, will have the burden of treating these problems, or at least approaching these problems, but also to educate your immediate and also your greater social surroundings. But in order to educate and obviously to medically approach these problems, you and all healthcare professionals will have to have a firm knowledge and the framework of how physiology of the reproductive functions in human works. And that's what we try to accomplish here, to give you a framework that you can use later in your study or as a doctor or any healthcare professional. So how it's done, which topics will we touch on? I'd like to give you an overview. First of all, in the introduction today, I would like to provide just a general overview of the reproductive system, how it works, which are the anatomical structures, which are the hormones that play a role here in both sexes. All right? Then we will discuss the male reproductive functions in more detail. And then we will see how during intrauterine life the primordial gonads and tissues decide which way to go, whether go down the male or the female type differentiation. And then Professor Speth will talk a lot about the female sex hormone and its reproductive functions, special emphasis on contraception, and at the end of the lecture series we will be hearing about pregnancy and physiology of childbearing in general. Today we'll try to cover these two topics. I'm afraid we will not be able to finish all the aspects of male reproductive function, so we will end the lecture on a cliffhanger. I will not talk about erection, so that you may want to come back after the spring break. And then in the next lecture, we'll try to cover those two, all right? I forgot to ask, but I hope that you had a chance to download the slides and have them with you. That'd be really desirable, because then it's easier to follow. My handwriting is just naturally awful, so you wouldn't be able to read it. So having the slides at hand makes it easier to follow, all right? And that also applies for the next lecture. Okay, so try to have the slides at hand. So what's the general layout and which are the hormones that are involved here? We're going to talk about the hormones in both sexes, and just to give you a technical heads up, at least in this slide, everything in green should indicate a positive or activation, a positive effect or activation, and the arrows in red should indicate inhibition, all right? Unless otherwise specified by me, that will be valid for all other slides too. This shouldn't be any surprise to you that, yet again, we are going to deal with the hypothalamus, pituitary, and peripheral organ axis. And in this case, the gonads will be the last player in the axis, the third player, the peripheral player, but again, the hypothalamus and the anterior pituitary will be heavily involved. As usual, we will have a releasing hormone, and that is the gonadotropin releasing hormone, or conveniently abbreviated GnRH. This releasing hormone, as usual, will be secreted by neurons into the portal system of the anterior pituitary. And the capillaries and circulation will carry or transport the hormone to the anterior pituitary, and more specifically the so-called gonadotropic cells in the anterior pituitary. And these cells, not surprisingly, again, they possess GnRH receptors, which is a GPCR. You know what a GPCR is? It's a G protein coupled receptor. This happens to be GQ coupled. No one will ask you this information, actually. And GnRH will activate the gonadotropic cells so that they release, this time not one, but two stimulating hormones. The first one is the follicle stimulating hormone, or FSH. I will refer to it as FSH from this point on. And the other one is the luteinizing hormone, also known as neutropin. We will refer to it as LH. So here we have a cell in the anterior pituitary which produces now not one but two hormones. We will later on discuss what are the differences, what are the similarities between these two hormones. As usual, these stimulating hormones will mainly activate the gonads. They will respond in two ways. First of all, they will activate and maintain normal gametogenesis. That could be the generation of new egg cells, or could be the generation of new sperm cells. The gonads will also produce a relatively wide array of hormones. These hormones will include androgens. I named one, specifically testosterone, because that's the main androgen in humans. The gonads may also produce estrogens. I named estradiol, or E2, because in women in fertile age, that will be our main estrogen. The gonads may also produce gestogens. And I named progesterone, P4, that will be the main progestogen in the fertile age in women. And some hormones are not steroids in their chemical structure, but rather proteins. I named here one family, inhibin or inhibins. They will have both local and systemic action. So the hormones produced by the gonads will have local actions, paracrine actions if you so wish. But also they will carry out, as usual, negative feedback effects on both the hypothalamus and on the pituitary. So that's how the regulation is actually a closed circle. And I also have to add that you will hear about these hormones in great details, that the anterior pituitary also will produce prolactin. It will be very important during childbearing, and oxytocin. That comes from the posterior, from the neuronal part of the hypothalamus. They have pleiotropic effect mainly in female, but also probably in male too, and also will alter behavior to some extent, also in humans. You'll hear about that later on. Okay. As I've already mentioned, steroids will play a huge role in carrying out effector functions in our reproductive system. So let me just quickly recap how they are produced, when and where they are produced. Okay? The issue we're about to discuss, it should be more or less familiar to you by now. Not surprisingly, the first step of steroid production is the side chain cleavage that yields us pregnenolone. You also may have heard that this is the rate-limiting step of steroid synthesis. So the speed or the activity of this biosynthetic step will have a huge effect on the activity of the total steroid production in the pertinent cell. We also know that hormones that activate steroid production in steroid-producing cells engage either the CAMP and PK pathway or the calcium pathway. So if there's a CAMP signal or a calcium signal, this first step will be boosted. Steroid production will go up. Then we have pregnenolone. Now the question is, what's the enzyme toolkit of this very cell, the cell in question? Because this enzyme toolkit will ultimately will be decisive which steroid product will be in that cell produced in that cell, all right? So pregnenolone can give rise to progesterone. You've already learned about progesterone as a precursor to other steroids, right? But also progesterone on its own right is a very important biologically highly active gestogen. In fact, it will be our most important gestogen in the female reproductive system. It's also abbreviated P4. You know these biosynthetic pathways. Progesterone can serve as a precursor for both aldosterone and cortisol. And pregnenolone can be substrate for the synthesis of dehydroepiandrosterone, commonly abbreviated DHEA. And these two, androstenedione and DHEA, are the most important androgens that are produced by the adrenal cortex. So what we have here is actually the model of the adrenal cortex. That would be the zona glomerulosa. That would be the, which one?
Fasciculata.
That would be the fasciculata. And these two hormones come from the reticularis. These two hormones are weak androgens. Alright? They will play an important role later on, mainly in female. Okay, let's go further. Beg your pardon?
Can you repeat the location?
This was the zona reticularis, the innermost layer of the adrenal cortex. That's in the one in the middle, so that's the zona fasciculata, and that is the glomerularis. Glomerulosa, sorry, I made a mistake. I made a terrible mistake, my bad. Not glomerularis, it's glomerulosa. Oh my gosh, I'm going to be crucified for this. So, these are weak androgens. What happens actually in males? We have a lot stronger androgens than these, haven't we? We do, and that happens in Leydig cells. The Leydig cells, as you will see later on in great detail, are able to produce a lot stronger androgen, and that is testosterone. Testosterone is the main androgen in humans, at least in males. Can be produced by these biosynthetic pathways. I would also like to bring it to your attention that gestogens can serve as a precursor for the synthesis of androgens. This will later play a role. Okay, we have testosterone. Testosterone in some organs locally can be further metabolized by the 5-alpha reductase enzyme into an even more potent androgen, and that is the 5-dihydrotestosterone. That happens almost exclusively in males, but will have important function on the prostate and the intrauterine differentiation. Testosterone serves as a substrate for the enzyme aromatase, also referred to as cytochrome P90. And the aromatase produces estradiol, which will be the most important estrogen in women during the fertile age, in non-pregnant women. Alright? The same enzyme can produce another estrogen, estrone, from androstenedione. This will be important in the placenta during pregnancy. And in the fetus, estradiol can be turned into estriol. It has 3 hydroxyl groups, and this will happen mainly in the fetus. It can be used as a biomarker of certain fetal functions. I will be given serious rebukes for showing you this one, but I really like this picture. It's readily available at Wikipedia, and I think this is the single best representation of steroid biosynthetic pathways. The colors, the coding, the enzymes are all there in a relatively easy to decipher way. Okay? So if you're so inclined, if you so wish, you can study this in greater detail. Actually, this is one of those instances where a little bit more will mean much less. So if you understand and know these biosynthetic pathways by heart, I don't want to say you have to, but I suggest you get acquainted with it. It will be a lot easier to memorize all these functions that will follow. Okay? And those are the commonly used abbreviations for your convenience. Okay? This is only a suggestion. So, how do these steroids in both sexes, still talking about both sexes, exert their effect? Now, there's the classical way, which we all know. They tamper with the gene expression. There are two ways to accomplish this. One is that I'm going to use estrogen as an example in these slides, but this could be any steroid, really. Estrogen binds to its receptor, goes to the nucleus and binds special estrogen-responsive elements in the DNA, recruits other co-activators or co-repressors of transcription, and then changes the transcription activity of the pertinent gene. There's another way that this could be accomplished where genes are actually waiting for the estrogen to come, where the activators of gene expression are already pre-assembled. When estrogen then eventually arrives, the receptor binds to this complex and turns on or off the gene expression. So these are the two ways that gene expression or transcriptional activity can be turned on and off. And I'd like to emphasize here that this way, the transcription of a gene can be increased or decreased depending on the gene, right? So we can either induce or repress in the same cell. Let me give you an example from your future clinical work. Let's take the effect of estrogen on hepatocytes, right? If we have increased estrogen in blood, for instance, because the female patient is on contraceptive pills, that would be a very typical example. The higher E2 activity will increase the transcription of two coagulation factors, namely 8 and 10. And at the same time, it will decrease the expression of antithrombin 3. Now, what will be the direct corollary of that? What do you think?
Coagulation.
Exactly. That substantially contributes to the increased risk of thrombosis if the estrogen levels are high for a longer period of time. So as you can see, in the same cell, genes can be turned on or turned off. There are other ways how steroids influence cell and tissue activity, and those are the non-genomical effects. These are really just to give you an idea of the versatility of steroid physiology. You won't need to know the next examples, but in the 21st century it's probably advisable to at least hear about the fact that some steroid receptors can reside in the plasma membrane. They can bind the steroid from the extracellular space and activate phosphorylation cascades on their own, or they can manipulate the activity of other receptors, and that way they can turn on and off the phosphorylation cascades and tamper with the transcription. Alright? That would be a quicker way to change cellular functions. And also there is yet another possibility. Some sex steroids have bona fide 7-transmembrane G protein coupled receptors, and estrogen would be such a steroid. GPR30, so G protein coupled receptor 30, is a 7-transmembrane receptor. It can readily bind estrogen and activate tyrosine kinases, or, interestingly enough, a portion of these receptors reside in endomembranes. And when estrogen binds to these receptors, mostly in the endoplasmic reticulum, calcium can be liberated, and we all know that calcium has a very versatile effect on cell function. Alright, so there are quite a few ways in which steroids can alter quickly or with a longer lag time the cell function. It also has to be added that most of the effects we're going to discuss will be through the alteration of gene expression. Okay? So, that was the introduction to the entire lecture series. And let's see how these general rules now apply to the male reproductive system. What can be achieved by using only these anatomical structures and steroids and hormones? So what's the hormonal background? What we're going to discuss here is the hypothalamus-pituitary gonad axis in fertile males. Okay, so that's after puberty. Once the system is stable and it's running as it should, because the axis will work a little bit different before puberty, as you will see next time. So we have the arcuate and the hypothalamus. We will have the gonadotropic cells as usual, and then we will have the gonad with practically two main cells carrying out functions. One will be the Leydig cell or Leydig cell. This person was a German guy, so we should probably say Leydig cells, but in the English literature sometimes it's referred to as Leydig cells. I will probably mix up these two versions. And the other one is the Sertoli cell. GnRH will be produced in the arcuate nucleus and will activate both LH and FSH secretion by the gonadotropic cells. LH will affect predominantly, not exclusively, predominantly the Leydig cell function, whereas FSH will activate mainly the Sertoli cells. They will respond to their stimulating hormone, the Leydig cell by producing testosterone, and the Sertoli cell by stealing testosterone from the Leydig cell and producing estrogen. Also, it will produce other factors which will be mentioned later on. One, in this aspect, will be important right now, is inhibin. So, what happens then? What do you think is the next step in this system? In the view of all the hypothalamus, pituitary, peripheral organ axis that you are now acquainted with, what should be the next step?
Negative feedback.
Yeah, we will have negative short loop and long loop negative feedback. In fact, no surprise, no fancy dancing. Testosterone will, obviously with the circulation, reach the arcuate nucleus. What's interesting though, that as soon as it enters the arcuate nucleus, it's immediately turned into estrogen locally. And then this locally formed estrogen will carry out the negative feedback on GnRH production. Alright? So there's an extra twist in the system, but only this little one. Testosterone, but also dihydrotestosterone, circulating in the blood, they directly inhibit the gonadotropic cells by dampening LH and FSH secretion. So that would be the long loop, that's the short loop. Additionally, inhibin, and also to some extent estrogen, but that's probably negligible in males, will inhibit the FSH production, specifically the FSH production. Okay? So that is our system. And by drawing these curved, crooked arrows, I wanted to suggest or symbolize something. Do you know what I meant to imply with these curved arrows rather than just drawing a straight line.
Pulsatiles.
Very nice indeed.
Pulsatiles.
Why am I here? Yes. So this is the GnRH concentration in the CSF, so in the liquor, in the cerebrospinal fluid, in a non-human primate. And that's important after a gonadectomy. So there are no sex steroids around. What we see here is the inherent intrinsic pulsatile activity of these cells, all right? They have no feedback from the periphery. Even without any information from the periphery, they produce GnRH in a pulsatile fashion. 2 days or 5 days after the gonads were removed. Now, in the view of this, if we were to measure, for instance, the LH or the FSH concentrations, what do you think we would see? Yeah, let's just measure the concentration right here, also in the liquor. Beg your pardon? The question was, we know that GnRH is secreted in a pulsatile manner, right? We see the peaks and it comes down, goes up. Very regular, I would say. Now, we measure the LH-FSH. What do we expect?
Also pulsatile?
Yes, we also, because they are directly governed by GnRH in the system without peripheral steroids, they also display a pulsatile secretory activity, right? Very nice. Next question, what do you think? How would the concentration look like, or what would we measure if the gonads were still present?
Low. Lower activity?
I also think so. That we would still see the pulsatile nature of the system, but since in this case we would have negative feedback, probably the peaks were a little bit smaller, right? But the pulsatile nature of the entire system should be still present. Okay? Now the question is, why aren't these episodes entirely rhythmic? Why isn't that absolutely regular? What do you think?
Desensitization.
Desensitization. So this is desensitized and then sensitized, and I cannot exclude, but I don't think that's the easiest. We don't have negative feedback, we don't have the gonads anymore. Beg your pardon? So there's a delay. I mean, if we had a delay, we always should see the same delay, right? If there's an inherent problem with the technique we use, that should only cause a frame shift. That's because we shouldn't forget that we're in the brain, right? So the cells in the arcuate nucleus, they don't only take into account, okay, what's the sex steroid level? They integrate so many other signals. For instance, light. If we stress out the animal, has the animal eaten? Is it in fasting? Is it in the fat state? Also, circulating leptin, insulin levels, emotional stress, glucose level. They are all integrated into this system. Okay. So even though we tend to forget that it's a fairly complex system and we only focus on the feedback loops, they are utterly important, obviously, but still we have to take into account that we're in the brain and the hypothalamus is a master integrator of lots of stimuli. Okay? And let me show you how the testosterone level in peripheral blood looks like during the entire lifespan of a healthy man. Okay? So this is the plasma testosterone concentration, and This is the age. You will have to take my word for it right now. We have two peaks early on in life. Once during the intrauterine development and one right after birth. We will not discuss these right now, but next time I will explain and we will see why and how these come to be. What we focus on right now rather is what happens here at the beginning of puberty. For all we know, this is where the GnRH secretion starts. And this GnR secretion, what will be the direct consequence? The direct consequence will be obviously that FSH and LA follow suit, right? Because they're governed, as we discussed previously, they start to increase their concentration and the gonad will respond to that. Now there is no runaway effect here because as soon as we turn on testosterone production, this will exert negative feedback on FSH and LH and the system achieves a new steady state where the FSH and testosterone concentration in the blood are fairly stable. Now this temporal resolution is not enough obviously to see small pulsatile changes in the FSH and LH, but we are not paying attention to those right now. So my question is, what do you think here at later ages of about 70 or 80, the testosterone production begins to decline and FSH and LH at the same time display an increase. Why is that?
Less negative.
Because the system is still sensitive for the negative feedback. Less testosterone means less negative feedback. FSH and LH go a little bit higher. This effect you see here will be a lot more pronounced and a lot more important, probably important too, in women after the menopause. And I think that we all deserve a break at this point.